All right, so I'm planning to finish chapter 10 on inheritance and DNA and lay the foundations for talking about microbial genetics and genetic engineering. So we have to finish talking about DNA, RNA, and what they're used for, which is to make proteins. So we're going to go into DNA and RNA structure, describe how DNA and RNA lead to expressed genetic traits. All right. So last time we talked about the evidence that suggests or strongly suggested that DNA was the genetic material, and we started in on DNA structure. And so this all led up over about a 70-year period to Watson and Crick identifying the structure of DNA. And DNA is a nucleic acid. Nucleic acids are made of nucleotides. There's two types, RNA and DNA nucleotides. All nucleotides contain a phosphate, a sugar, and a base. In DNA, the base or the sugar is deoxyribose. So it's a 5-carbon sugar, forms a ring-like structure. And then the bases are shown in this next one there. 2 pyrimidines, cytosine and thymine, and then 2 purines, adenine and guanine. Okay, and so with phosphate and deoxyribose, each nucleotide contains one of these bases. The nucleotides are linked by these phosphodiester bonds. These are covalent bonds that link phosphates and sugars together to form a backbone, and then the bases are exposed in each chain. This is a real durable thing. These are covalent bonds. This won't fall apart. The key thing with DNA is that DNA contains 2 backbones, 2 chains that are matched together and held together by these weak bonds between pairs of bases. Adenine and thymine form 2 hydrogen bonds that cause them to link, and then cytosine and guanine form 3 hydrogen bonds. Okay, and these link 2 strands of covalently linked nucleotides together. And so wherever there's a thymine on one strand, there will be an adenine on the other, and vice versa. And wherever there's a cytosine on one strand, there'll be a guanine on the other, and vice versa. These hydrogen bonds that hold the bases together, these are weak bonds. They're not permanent, and they allow the strands to be separated if need be. The strands can be separated, and that happens sometimes when they're copied, to copy parts of them when they're making proteins, or to copy whole strands when DNA molecules are being copied, when cells are getting ready to divide. And this is kind of a key thing, that these complementary base pairs can be pulled apart. And if you have the sequence of one strand, you know the sequence of the other strand. And so that's kind of a key thing. As soon as Watson and Crick figured out the structure of DNA, they figured out, okay, this is how the strands are copied. And then they also figured out that the strands could be partially separated and put back together inside the cell. They can be split and rejoined, and so that allowed short segments to be used whenever the cell needed them to be used. And so that was a key thing as well. At the same time that the structure of DNA was being determined, the structure of RNA was also being determined. And RNA is ribonucleic acid. It's just slightly different from DNA. It's also made of nucleotides, but in the case of RNA, there's a different sugar. The sugar is ribose instead of deoxyribose, so it has an extra oxygen atom. And then the pyrimidine uracil replaces the pyrimidine thymine in RNA. And these seem like minor changes, but it means that RNA almost never forms double-stranded molecules. It forms single-stranded molecules that sometimes form loops because of base pairing, but it never forms entirely double-stranded molecules. And so RNA exists in different a different form, and it's generally found in different parts of cells doing different jobs. Whereas DNA molecules are kind of, in eukaryotic cells, DNA is in the nucleus and kind of serves as kind of a library of possible things the cells can make. RNAs, once they're made, they're involved in converting instructions from DNA into proteins. Ribosomes, the most notable thing they make are, or RNA, the most notable thing RNAs make are ribosomes. And ribosomes are involved in taking messenger RNAs and converting them into proteins using transferRNAs. And so we have several different types of RNAs, and RNAs kind of act as enzymes. They have specific jobs. They form and break bonds. They work with other things. They're quite active. Perhaps the most common RNAs are transferRNAs, and these carry amino acids to the ribosome, and on the other end, they actually read the genetic code. And so here they show a messenger RNA that has a sequence, and the transfer RNA can read a 3-base sequence by having a complementary sequence. And so here the RNA has a sequence of GAG. You can see that kind of at the bottom there. And then the transfer RNA will have a complementary sequence, CUC. And wherever this is found, this sequence is found on a messenger RNA, the transfer RNA will bring an amino acid, here glutamic acid. So this is one diagram, this is another diagram. Okay, and so really cells have a fairly simple genetic machinery. Their DNA stores the code for making proteins, okay, and the RNAs use that code to assemble proteins. And the 3 types of RNAs involved are the ribosomal RNA, the messenger RNA, and the transfer RNA. And this is primarily what all cells do, is they're all making proteins, but they can make different proteins depending on what the code they have. Okay. In addition to that, cells can kind of regulate what proteins are produced and when they're produced. And so like here, this is a Serratia marcens. It's a microorganism that we sometimes use in the lab, and it has a gene for a pigment, but it only makes that pigment when temperatures are cool. If it's warm, it doesn't make that pigment. And so here you have the same organism that can do different things at different times depending upon when the cell activates its genetic material. Okay, and this is typical of cells, is that they have parts which we call genes that are made of protein, and these are separated by areas in the DNA that never are made into protein. We call them non-coding DNA or non-coding regions, but that are often regulatory. They determine when the gene is actually made into a protein. So they're regions that can be important too. Okay, so all cells basically kind of work the same way. All cells have DNA. Okay, and in eukaryotes, this is always in the nucleus or in mitochondria and chloroplasts. Okay, and that DNA has to be copied when the cell reproduces, and otherwise its only job is to direct RNA to make proteins. Okay, in prokaryotes, it's similar. Prokaryotes have a single chromosome, but that chromosome is doing the same job. It's storing genetic information, and it's directing the production of proteins in the cytoplasm. Prokaryotes sometimes have some additional pieces of DNA floating in the nucleus or floating in the cell. These are called plasmids, and they contain genes for sometimes some important thing like resistance to antibiotics or a toxin. Okay, so all cells use their DNA to make RNAs to make protein. Okay, even the simplest bacterial cells, which may have several million base pairs, are making proteins. And even the most complex cells of plants and animals, which have lots of DNA, maybe 1,000 times more, they're doing that same thing. And so it was really a surprise to figure this out, and a surprise to find out that they all use the same genetic code. But that's one of the great mysteries that's been solved, is that we know how heredity is passed from parent to offspring, and how traits go from stored information into physical traits. And so that's one of the mysteries solved.